If the bee disappeared off the surface of the globe, then man and woman would only have 4 years of life left. No more bees, no more pollination, no more plants, no more animals, no more man or woman. Albert Einstein. What would Einstein possibly know about bees? Can Einstein be trusted? Actually, I think he can, and bees are tremendously important. And that's what is going to be really the vehicle that takes us through this review lesson. And so the review is about how to design an experiment. But it's really important to understand what's going on with the bees right now, too. And so another thing to just keep in mind is on the AP exam, I think environmental services is going to be a really important thing to have ready to go at the tip of your tongue or the tip of your fingers. And pollination is a really important environmental service, one that we won't appreciate until it's gone. And so, objective, IPM is important. What is IPM? We're going to get into it. And how does it relate to the bee colony collapse disorder? And so maybe you've heard of the colony collapse disorder. In past years, I'm always trying to get strategic about what the FRQs are going to be about. And I always think it's going to be this, and it hasn't been. Maybe this is the year. Especially since, I don't know if you can see the date there, it's 2020 January. Colony collapse disorder caused the loss of an estimated 40% of honeybee colonies in the U.S. last year, the worst winter decline on record for tracked hives since research began. And so colonies have been on the decline for a long time. Just to skip ahead, this is like the latest. Scientists modify honeybee gut bacteria to fight colony collapse, causing varroa mite and deformed wing virus. And so, how cool for biotechnology to adjust a bacteria that goes into a bee to help them fight a virus. Maybe we consider doing something with that around COVID. Who knows? But that's another topic. Colony collapse disorder is a pretty crazy thing. Here's back in the 50s. These are the bee colonies that they've been keeping track of. And of course, you can notice the decline until we get here is when they really started to kind of freak out about it. Where you got the CDC colony collapse disorder from 2007. Here's another graph that goes up to 2017, and of course that newspaper article, 2020, worst winter ever. It's happening. What is going on? And of course this is what scientists are trying to find out. Here are some hypotheses. Is it climate change? Is it over pesticides, habitat destruction, or is it a parasite or virus? Answer. Probably a little bit of everything. It's a complicated, multifaceted issue, and it's getting exacerbated by different factors. I want to show you just a few minutes of this video here that takes place right in our backyard in UC Davis. There's a little bit of San Francisco here too. I'm just going to show you the first few minutes to kind of get an overview of colony collapse disorder.
In 2006, scientists came up with a name for the mysterious, troubling bee die-offs they had been witnessing throughout the United States. They called the phenomenon colony collapse disorder, CCD. The die-offs have continued unabated, threatening billions of dollars of California crops, from sunflowers to almonds. Each February, beekeepers truck more than half of the country's honeybees to pollinate California's almond crop, worth $2 billion. But roughly 1/3 of honeybee colonies have disappeared each year since the crisis began. Beekeeper Randy Oliver had it happen to his hives.
With the colony collapse, it's very sudden, where you see the colony just kind of teetering, still full of bees, and then boom, overnight, all the bees are gone.
Scientists now believe that a combination of pesticides, mites, poor diet, and long trips across the country are making honeybee colonies vulnerable to pests and pathogens. But the specifics remain a mystery.
One of the frustrating things with CCD is it doesn't look like there's any one single agent or culprit that you can point the finger to. Imagine if you had the cold and you got the flu on top of a cold. Well, that might be the case with the honeybees. You have a weird fungus and the virus, and it causes a drop in the health of the colony to the point where the colony can't maintain itself. There's no gentle way to get in there.
At the University of California at Davis, scientists are trying to find ways to improve bees' health, partly by changing what they eat, partly by breeding hardier bees. Eric Musson has been the University Extension apiculturist throughout the colony collapse crisis. Musson is frustrated that the disease is still rampant.
What I was hoping originally was that this was going to be sort of a viral epidemic. It was going to come in just like flu comes through a human population. The susceptibles are gone, but in a couple of years it just sort of disappears and you have to wait, hopefully a long time, before the viruses mutate and come back and create a problem. But this one's not going away. This one's entrenched, and that's not good.
In their search for answers, scientists have developed some innovative tactics. At the University of California, San Francisco, researchers are using tools developed for studying human pathogens to hunt for culprits in colony collapse disorder.
In comparison to what we know about human biology and infectious diseases that affect all of us, we know almost nothing about honeybees, for which we depend on so much. 1, 2, 3.
So scientists are essentially starting from scratch. Using a modified vacuum cleaner, they collect healthy bees from nearby hives to try to figure out what pathogens normal bees contain, so they can recognize abnormal when it occurs.
Honeybee colonies, kind of like human populations, are exposed to a number of viruses and pathogens throughout the entire course of the year. So what this study provides us is a normal, healthy colony baseline of the ebb and flow of the microbes associated with that colony throughout the course of the year.
In the lab, researcher Charles Runkle smashes up the dead bees to extract DNA or RNA and analyze what viruses or bacteria are present. As part of the study, the scientists also followed and got samples from a huge commercial beekeeping operation as it traveled across the country, pollinating crops.
Hit stop. Who knew about the bees, right? How interesting. And what a complicated just situation and the stakes are high. And so people are doing all kinds of experiments, and again, that's what this, I guess, little flip lecture is about. And so we're going to shift gears just a little bit here to Monsanto. And Monsanto, of course, is a huge biotechnology or biogenetically modified foods corporation. And they are famous for selling Roundup and a lot of other things. And personally, I think that there is room for genetically modified foods, and how, can we grow as much food as we can with as little amount of energy input and as little amount of habitat as possible. I think we need to be open to that. I do have qualms with one company doing it all, like Monsanto. And now Monsanto has got some, there's some evidence that it might be linked, the Roundup is linked to cancer. It's a huge corporation, there's a battle, and these are recent headlines around it too. And there's more people that are signing up. One thing to note is that all of the lawsuits, there's a judge in California that's kind of green lighting all this. And so a lot of the press is coming out of California. Not sure what that means. But anyway, Roundup is, pesticides are used all over the place, or ubiquitous. So just picture that you are a farmer, you're a hydroponics, aquaponics farmer, and your livelihood is tied to that. And then all of a sudden you got all these little buggies showing up on your plants, and they start eating everything. They start eating your livelihood. So back in the day when in like the 50s, when pesticides first became popular, farmers were all in, of course we're going to use it. Let's kill the things. And it made total sense. And it wasn't until Rachel Carson came out with her book that really started to raise the consciousness. Silent Spring, hopefully you've remember hearing of that, that these pesticides can get in the food chain and they work their way up. And remember, we've studied a little bit about birds and the bioaccumulation, biomagnification. And so anyway, this leads us to IPM. What is IPM? Take notes on this. IPM stands for Integrated Pest Management. And so the idea of this is we're not just going to use chemicals anymore. there's a place for chemicals, there's a place for some spraying, but we're not just going to bomb everything with as much chemicals as we can, because we know that those chemicals can have unintended consequences. And so what else can we do besides chemicals? And so there's a variety of things you can do. And if you remember, we had we did some flip lectures on polycropping. And so in the United States, typically we just grow one thing, miles and miles of corn or wheat, and that allows for bugs to come in and just go from plant to plant to plant. And so maybe we mix that up. Maybe we introduce ladybugs or cane toads, for that matter, but you've got a biological control that can come in. Maybe you can mess with it. I mean, all different kinds of things. What about loud music? What about spraying with hot water or soapy water? There's all different kinds of things we could do. So here's what I want you to do. And so again, integrated pest management is you're using a little bit of all these different strategies, right? So you can do a bunch of different things to control the pests. And so you're the farmer and you got your crop is getting hit by these aphids, which are a common pest. Hit pause and jot down 5 things you could try besides pesticides to control your aphid population. Hit pause. Okay, and now we're back. And so what we're going to do now is we are going to design an experiment. And this is something we're going to enter into the practice AP exam by the College Board. And so your goal is to reduce the use of pesticides. How? By a combination of chemical, physical, and biological methods. And so here's just a couple ideas that I brainstormed. Could spraying bugs with soapy water work? Could it be that easy? Remember, soap can clog the membranes. Bugs breathe through their exoskeletons. And so here's what I want you to do. I want you to hit pause again. We're going to design an experiment, and you want to have these things. You want to have a purpose that identifies the independent and the dependent variable. That the dependent variable, of course, is what the data you're collecting. The independent variable is the variable you're manipulating. I'm going to play gangster rap really loud. There's a control. I'm going to point out the control. The control's not going to get the treatment, and then maybe I'm going to have different volumes of gangster rap, or maybe different artists too. Some more effective than others. I'm going to write a hypothesis. Doesn't have to be an if-then statement, but if you can do it, it's great. And then I'm going to come up with step-by-step methods. For sure, I'm identifying the control. I'm talking about my sample size, the number of brine shrimp that I'm testing in my LD50 experiment. I'm going to say how long they're going to be exposed to the treatment. And then eventually I'm going to say what results I'm looking for. After 10 minutes, I'm looking for the number of dead brine shrimp. Or, you want to be as specific as possible with the results. And then with extension, here's a next great step. We're going to repeat the experiment. We're going to replicate the experiment, 1,000 times to make sure that this data is good. So go ahead and hit pause and write up your experiment. And actually we can just be complete right now, but we are going to do something with that written up experiment. So you've got your notes on integrated pest management, hopefully a little bit from the bee colony disorder too. And now you've got an experiment that you're writing up with those things in it. We're going to be as specific as possible, and then we're going to put that into the College Board thing. I'll post an assignment for that in Canvas as well. Okay, you guys, we are going after it. Everything we can do right now helps. All right, take care.